The overall goal of this experiment is to analyze bacterial growth precisely in a high-throughput manner. Using this procedure, either multiple media or varied strains can be tested simultaneously.
This method can help answer key questions in the systems biology and the microbiology fields, such as what determination factors of cell growth are. The main advantage of this technique is that it allows researchers to precisely evaluate global parameters of maximal growth rate and maximum population density in growth curves of bacterial cells.
Though this method can provide insight into E. coli growth, it can also be applied to other microorganisms, such as the growth assay and screening of environmental bacteria.
To begin, place 5 sterilized glass tubes with silicone rubber stoppers, pipettes, and the target strains on a clean bench. Expose the mouth of the glass tube to a Bunsen burner before opening the silicone rubber stopper. Expose the silicone rubber stopper to the flame after the tube is opened, and then lightly place the cap back on the glass tube. Next, use the electronic pipette and disposable serological pipette to add 5 ml of M63 to one of the glass tubes and 4.5 ml of M63 to the other 4 tubes. Use the P200 tip to pick a colony and inoculate it in the glass tube containing 5 ml of M63. Vortex the tube to make a suspension. Then, dilute the solution tenfold by transferring 0.5 ml of this solution to one of the 4 tubes.